individual informations, we found that there was very high population redundancy. Then, a second way of asking whether the correlations matter is to do these decoding tasks. So you have responses of neurons, they're correlated. It might be the case that you could still build a linear classifier that ignored the correlations and did a good job. That's conceptually possible. There are some cases that people describe in the literature where that can happen. But what we found here is that when you look at the full population, it makes a huge difference to know the correlation in order to read out the information well and get into this very low error limit. So, roughly speaking, what we see for all the ganglion cells is that they have these kind of broad tuning curves. Okay, so that means that when you consider all the possibilities that you have for the visual stimuli, the responses are pretty ambiguous. And then so you need a large population to reduce the ambiguity. And there's kind of extreme functional diversity, and so the basic kind of picture that we have is that there are going to be visual stimuli, let's say, that are behaviorally important, like maybe becomes important to recognize the letter X. Okay, you don't have any individual ganglion cells that are X detectors. The information that a spatial pattern like that has occurred is represented in the firing of some cells and the silence of other cells. You don't really know who those cells are going to be ahead of time, because their response functions are quite diverse and quite complex. And so basically, the brain is going to have to learn who fires and who doesn't when there's a particular stimulus that becomes important to recognize. Okay, so I think this population code is fundamentally one in which subsequent circuits are going to have to learn. Like it's just not possible to kind of know ahead of time by knowing things about the cell type, who fires and who doesn't fire when there's some arbitrary stimulus like the letter X. Okay, so I've told you that the retina has a collective code. That's another way for saying that the correlations matter, and we can't kind of look at each neuron one at a time. But what I think is really exciting here is that the multi-electrode array technology is such that we can, we think we're on the verge of witnessing the entire population code. And just to put that in perspective, this is completely impossible with current technology for any other part of the brain, essentially. If you think about area V1, there are 10,000 neurons in a hypercolumn, and you can't even come close to looking to recording from all of them simultaneously. So I think the retina is really a great system for understanding this type of issue of how the collective activity of multiple neurons represents information, because we can really measure the whole thing. And again, it's kind of particular patterns of spiking and silence among the ganglion cells that identify particular stimuli. And so there are also many ways in which I think the retinal code is similar to population codes in other parts of the brain. In particular, this pattern of kind of weak correlations, usually positive, so cells more often fire together than not. Kind of weak positive correlation shared among many neighbors is something that is certainly seen all over sensory cortex and other parts of the brain. And so again, there's the hope that things we figure out first in the retina will help us understand population codes in other parts of the brain. And so with that, I'd like to thank various lab members who contributed to this work, and thank you for your attention at this late hour of Friday.